Hello, my name is Andy Waters and I'm Professor of Developmental and Molecular Parasitology here in Glasgow. I'm also Director of the Wellcome Centre for Integrative Parasitology and a Wellcome Principal Research Fellow. I want to spend a few minutes explaining on what our group works as a general background and then explain in more detail the principal project that we are offering as part of the IIB PhD programme. Of course, if you have ideas that you think fit well in our research portfolio, I would be delighted if you wish to discuss these and evolve a project more of your own design. This image is a false-coloured scanning electron micrograph showing a female malaria parasite gamete in pink that has been produced in vitro and that is being engaged by a male parasite in blue. The male is still actually developing and producing 8 gametes, only one of which is required to fertilise the female. You can see the main body of the male at the base and behind the female, and if you count carefully you can see all 8 gametes emerging, and some have already stuck to the female to initiate fertilisation. Gametes are produced in the mosquito midgut by gametocytes that have their own origins in the mammalian host and are taken up by the mosquito as part of the blood meal, and a new environment stimulates gamete formation. Gametocytes, therefore, are the agents of transmission, and understanding their production and biology could lead to approaches to either prevent their production or make them ineffective. Both outcomes could reduce or even block transmission and reduce the burden that malaria imposes upon low and middle income countries in the tropics. Therefore, work in our laboratory is focused on understanding the very early events in malaria parasite transmission and has 2 principal questions. 1. How does the parasite commit to making gametocytes? 2. What and when are the early events in gametocyte differentiation, including determination of gender? Our laboratory was one of the discoverers of the transcription factor AP2G, the production of which is absolutely essential for gametocytogenesis. AP2G initiates the transcription of genes that lead to the irrevocable production of male and female gametocytes. Expression of AP2G is the first event in the only developmental decision the parasite makes in its entire life cycle. Everything else is a progression or cascade. AP2G binds to specific DNA motifs in the promoter region of genes, including its own promoter, thereby operating a positive feedback cycle. The extent that a malaria parasite population commits to gametocytogenesis varies and is influenced by the environment of the parasite. Expression of AP2G is also controlled by epigenetics and sits in an island of facultative heterochromatin, which can oscillate between open and closed conformations and control whether the cell enters sexual development. What controls the activity of the chromatin regulators is unknown and part of what we want to understand using our rodent model of malaria Plasmodium berghei. Such knowledge might give us an idea of how the parasite responds to its environment. To pursue this question, we have identified a mutant parasite that lacks a predicted antiporter and makes very few gametocytes. Effectively, this mutant does not respond to environmental changes. Our hypothesis is that this antiporter transports a key small molecule that serves as a regulator of gametocytogenesis. Therefore, using metabolomics, we wish to identify the substrate for this transporter, comparing wild-type parasites and the mutants that fail to express the transporter. We will use a full battery of genetic tools and molecular approaches to the analysis of the mutant in order to identify the substrate and learn more about control of gametocytogenesis. Ideally, we will be able to design mimic substrates as inhibitory drugs that can be used to reduce or eliminate transmission of malaria. If you would like to learn more about this project or speak to me about the design of a research idea within the questions that we pursue, please contact me directly at andy.waters@glasgow.ac.uk or 330-8720, which is the office number. Thanks.